Good morning and hello everyone. So today is going to be our last class for this semester. We're almost there. Our final is on Thursday, 9:50 to 11:50. Unless you have a DRC requirement, then it will be longer. Your final will be 75 questions to all multiple choice questions. So those will be 2 points each, total 150 points. Okay. But that's all for the announcements, and our last topic is going to be development and differentiation. So we will talk about differential gene regulation during development, and this lecture is based on parts of several different chapters from your book. So chapter 19, chapter 20, 21, mostly 21, but there are parts from 19 and 20 as well. Okay, so we have been talking about gene regulation of gene expression throughout the semester, and we've talked about this, that our genome itself is invariant. That means that's a stable, that's all cells have the same genome. However, the expression is different depending on the cell type. So different cell types, different tissues, different organs will have different expression of genes, different gene expression, and that will lead to different protein formation, different translated product, and those proteins will carry out different functions. So the body plan of all organisms, so multicellular organism, is quite complex. So this development of this complex body plan requires interaction between many genes, and all of these must be regulated. Otherwise, there will be problems. There will be developmental or physiological issues if these processes aren't very well regulated. So all organisms need, any organism to be viable reproductive organisms, all of these different genes need to cross-talk, to talk to each other, and must be regulated in a very controlled way. A typical animal genome, and that's from the smaller organisms or lower eukaryotes to even humans, they all contains approximately 20,000 genes, give or take. So it usually varies from about 16,000 to about 23,000. So a median is about 20,000 genes for all organisms. So differential gene expression that we have talked about throughout this semester, that has been a theme, is the synthesis of a protein or RNA, if it's a non-coding gene, in a subset of the cell comprising an embryo or an adult. So these subset of cells can be a different tissue, or it can be a different group of cells in the embryo that will give rise to a tissue. Okay. This differential gene expression of our invariant genome is the key to animal development, because this need to be regulated very well. Enhancers and other transcriptional control elements, these provide temporal and tissue-specific gene regulation. As we talked about, these specific transcription factors they regulate the gene expression in tissue-specific, cell-specific manner. These enhancers can bind to specific transcription factors, or they themselves can be produced in a specific tissue under specific conditions. So these enhancers, they can enhance an expression of a gene, or they can in a different way, an insulator can decrease the expression of the gene. So all these different components that we talked about, they work together to regulate the expression levels. It has to increase or decrease or stay stable. That all need to be determined and controlled. There are several translational and post-translational control of protein amounts or activities. We didn't go in a whole lot of details of post-translational modifications of proteins, but keep in mind it might appear that we don't have very many proteins because we have 20,000 genes, and if each gene makes one protein, that's 20,000 protein. But the genes can be they can have splice variants. They give rise to different proteins. So it doesn't have to. So 20,000 genes don't just make 20,000 protein. They make more than 20,000 protein, depending on the smaller protein or larger protein or protein made in a different way because of the splice variants. In addition to that, post-translational modification of proteins is very important as well. Post-translational modification usually happens in endoplasmic reticulum and Golgi body after the translation is complete. So that's why it's called post-translational modification, and most of these post-translational regulations are addition of carbohydrate or lipid. So a lot of glycoprotein formation is very, very important. So you might have heard of N-linked glycosylation. So those putting these different carbohydrate moieties at different places in the protein that makes that protein different from the protein that doesn't have those carbohydrate moieties. So just keep in mind that post-translational modification of proteins that we didn't talk in this class are also very important for regulation of expression or carrying out the function by these proteins. So how do we know that differential gene expression of an invariant genome is the key to animal development? So there have been a lot of different experiments, and now we know this for sure. But in the beginning, when we were still scientists were still trying to figure out that every cell has the same genome. If you look at if you extract genomic DNA, you can extract from any tissue, any place, the whole organism is going to be same. But when you isolate RNA, or if you look at the mRNA levels in different tissue, that will be different. So why is this? How did we learn about this differentiation, this different gene expression of the genome? So in 60s and 70s, there was the first clone, the Xenopus laevis. And what scientists did in this first cloning experiment, because the hypothesis was the nucleus contains all the information, the genome contains all the information. So if you take that nucleus from early embryos, it should have all the information in it. And if you put it in an embryo, in a differentiated cell, that differentiated cell will become a pluripotent cell. In 1996, it was really the big mammal was cloned, and this started the biotechnology. This is the rise of biotechnology industry, was cloning of Dolly, the sheep, where scientists showed the first time that we can actually make this whole organism from a cell. And what they did here was they had these two different sheep. This is Scottish blackfish cytoplasmic donor, and here's the Finn Dorset nuclear donor. So this is they took the cell, the somatic cell from this sheep here, and that is you here, and they enucleated it, so took out the nucleus. Here they took the nucleus from mammary cells, and this nucleus was then inserted into the cell from the Scottish blackface. Now this is the cytoplasm came from here, nucleus came from here. This is it's inserted by direct current pulse, so you can see the electric current, and here then the cell was grown in a media, in a petri dish, up to the blastocyst stage. So after this blastocyst stage, there was a surrogate ewe. That's where it was implanted, and Dolly was born. So from that one cell, the cell from one organism and one individual, nucleus from another individuals together gave rise to this to cloned this lamb, Dolly, and that was a big thing. Now that proved that our the embryonic cells can be used, can have all the information potential to make a whole organism, individual. Now, most mammalian embryo contain a small group of cells. So the embryo, if you think about embryo, and we will talk about it, this is called the inner cell mass, or ICM for short. This inner cell mass, it forms all of the tissues and organs of the adults. It's pluripotent. Now, totipotent is different from pluripotent. Totipotent is the zygote. The zygote has information to make all kind of cells, the whole organism. That's totipotent. ICM can make most of the tissue, or all of the tissues and organs. So about more than 200 types of different cell types that we have in our body. Those are formed by these inner cell mass, or ICM. So these are pluripotent. They can produce many cell types. The formation of ICM cells, these inner cell mass cells, it depends on 3 main sequence-specific transcription factors. So here, this figure shows more than 3, but these OCT3/4, SOX2, and NANOG, these 3 now we know are the most important. These are required. The others, these are more they're not obligatory. They're more facultative. If they're present, it's good. If they're not present, the presence of these 3 different ones can lead to formation of totipotent cells, or induced, not totipotent, I'm sorry, pluripotent cells, or induced pluripotent cells. So here, what this figure shows is this is somatic cells. That means these are differentiated cells. So if you take these differentiated cells and you transduce this by a combination of factors. So these are the factors, are these transcription factors. If you increase the expression of these transcription factors in these already differentiated cells, in differentiated cells, these specific transcription factors will not be active. These will not be expressed. These will not be working. But if you make these, you induce these cells with these, then these will become pluripotent cells, or induced pluripotent cells. These induced pluripotent cells can give rise to all 3 different layers, different levels of the embryo: mesoderm, endoderm, and ectoderm. And all these cells, ectoderm cell, endoderm cell, mesoderm cell, will give rise to different tissue type, different organ type. Okay. Let me go back. Let me play this video. I hope we can. There.
In this video, then, we'll look at the beginning of development, looking into the ovary at an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here, millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring. And this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the 3 beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns, and appendages start to bud out, You see the head forming in the eye, and then eventually we get a small embryo, and some months later, of course, this would be born as a young baby.
Okay, so I just wanted to show you that inner cell mass formation and all these three different layers that will give rise to different tissues. This very early development is conserved among all organisms. So all organisms, whether it's a placental mammal or it's a non-placental, say, bird, they all have very similar these developmental stages. So the embryo will there's going to be an egg, going to be a sperm, they will fertilize. So all fertilized eggs, if we're talking about here, all diploid organisms. So there's a haploid egg and haploid sperm. These will fertilize, will make a zygote. This zygote will go through multiple different divisions, nuclear divisions. Those divisions can now be different for different organisms. In some organisms, nuclear division happens at a faster rate and cellular division happens later. For example, it's very common in insects. In humans, nuclear division and cellular division happen at the same time. The nucleus will divide into two, and thus it will also divide into two cells. The cellular development will follow the nuclear division. But in all organisms, all diploid organisms, this is the scheme where the zygote, the fertilized zygote, the diploid zygote will divide into 2, 4, 8, 16, 32 cells, and will make this inner tube, that's inner mass of cells, called this inner cell mass. That's very important. So now if we just wanted to make sure that we cover all these terms in the beginning of this lecture. Embryonic stem cells, when we talk about embryonic stem cells, we talk about usually two types of stem cells: totipotent, which is whole or total. That's what toti means, is total. So the zygote is usually what we are talking about when we are talking about the totipotent cells. So zygote can give rise to whole organism, including the placenta or the associated structures. Pluripotent means plenty, several. So this means pluripotent. When we are talking about pluripotent stem cells, we're talking about all the cell types that are derived from this inner cell mass. So inner cell mass can give rise to any of the over 200 cell types in our body. So these are pluripotent cells. Together, this whole zygote is totipotent. There's some adult stem cells. There's some cells are stem cells in adults as well. For example, bone marrow cells. These are called multipotent, so they can give rise to a few types of cells. They are not pluripotent, they're not totipotent, but they're multipotent. That means they can give rise to multiple different types, but not like pluripotent cells that give rise to all kind of different cell types. These their specialization potential is limited. That's why these are called multipotent. And a good example is mesenchymal stem cell that give rise to bones, cartilage, and connective tissue. So these are the difference between embryonic and adult stem cell. There's different terms of totipotent, pluripotent, and multipotent. Okay. There are three major strategies for differential gene expression when we're talking of development and differentiation, cell development. These three strategies are cell-to-cell contact, mRNA localization, and signaling through diffusion of a secreted signaling molecule. So that's, I'm not going to talk about the third, the signaling through diffusion of secreted signaling molecules today, because we have already covered this when we talked about signal transduction. Because in signaling through diffusion means when there is there's a molecule that's diffused through the cells, and that it can find its receptor and bind to that receptor. That could be a hormone, that could be a peptide, that could be any small molecule that comes and bind to its receptor, and then it will initiate a number of different downstream processes. So what we are going to focus on today is cell-to-cell contact and mRNA localization. Okay. So as I said, signal transduction can respond to a variety of different triggers that originate outside of the cells, and these triggers can come to the cell and bind to the receptor. These can diffuse to the target cell, but some of these signals that require cell-to-cell interaction that's what we're going to focus on right now. So one of the good examples of cell-cell interaction is the neural cell formation in insects. So here this is an example of I think it's a Drosophila, because most Drosophila are model organisms, very well studied. So a lot of our example come from the model organisms. So in for the neural cell formation in insects require cell-cell interaction. There is no long-distance diffusion. They need to be right next to each other. So cell-cell interaction to ectoderm, into epidermis or neuronal tissues. So this is a neurogenic ectoderm, which is shown here. This is the embryo, developing embryo, surface ectoderm, neurogenic ectoderm. And these are the cells all around. So some of these cells will become neural cells. And how these become neural cells, how these are predestined to become neural cells, we will talk about that. So here is the lateral inhibition of surrounding cells. So if this cell is destined to be neural cell, it will inhibit all the cells around it to become neural cells. So this cell, it will inhibit all of these. Once this cell start becoming neural cells, all the others will stay as ectodermal cell or epidermis. So here this is neuroblast, ganglion mother cells, neurons are formed, and the rest of the cells stays as epidermis cell. So it's an insect progenitor ectodermal cell. They can differentiate to form the epidermis, or they can differentiate to form ventral nerve cord or neurogenic ectoderm, which is an internal structure here. So how it happens is there is a signaling molecule on their surface. The cells that are destined to become neuronal cells, they have this signal molecule on their surface called delta. So here, this is the neuroblast cell. So that means this is the cell that will become a neural cell. So they have these molecules here. This is called delta. These delta will bind to the notch on the epidermis cells. These have these epidermis cells. So here, if you look at this is neuronal repressor gene, notch gene target. So this repressor is off. This is all these other proteins are bound here at the promoter region. So this delta binds to receptor on the skin cells or epidermis cells, which are notch. So this is delta, and the cell that is destined to become a neuronal cell is the only cell that will have delta, and these have these notch. This delta will bind to notch here. Once this delta and notch bind to each other, this binding will activate the notch. So this activation means this delta will cause proteolysis. So activation of notch receptor by delta will cause this proteolysis. It will release a nucleus targeted domain called notch IC. So this notch IC will then move to the nucleus of these epidermal cells. So in these epidermal cells right now, the repressor is off. So that if repressor is off, that means they can be activated to become neural cells. But this domain, the nucleus targeted domain notch IC or intracytoplasmic domain of notch, that will go. This is proteolysis. This will go and this will move to the nucleus. Once it moves to the nucleus, it will bind to this SUH. So once this SUH, these other molecules cannot bind here, and this will activate the repressor. So that means it will make a repressor protein. That means this repressor protein will now stop them from becoming they will repress the expression of the genes that are required to become the neuronal cells. So this notch IC associated with DNA binding protein SUH. This notch SUH complex activates the gene that encodes transcriptional repressor. This will block the development of neurons, and these cells will become now epidermal cells. So the delta, these are produced from neuronal destined cells. These tell the other cells around them to not become neural tissue. They will say that, oh, I am the one who am going to become the neural tissue. You guys, please don't become neural tissue. You stay as epidermis. So this is a short distance signaling. There is no diffusion. This is because of these molecules that are on their cell surface. So these are cell-cell interaction. These cells are right next to each other, interacting by these different molecules on their surface. Second type is mRNA localization. This mRNA localization is where and which mRNA is localized in the embryo. So the mother ovary makes some of the mRNA and put them in the embryo before that embryo is fertilized. So becoid or bicoid, and anterior structure commitment in Drosophila is due to mRNA localization. So here in Drosophila, in most organisms, the embryo will have an anterior pole and a posterior pole. When I say most, well, all will have an anterior pole and posterior pole, but it is easy to see in some eggs than the others. You can see the anterior and posterior pole pretty well in a chicken egg, because there's one side is narrow, one side is the posterior is the wider side, the anterior is the narrower side. So you can tell that from some eggs, but other eggs are more spherical, so you can't really tell which one is anterior, posterior, but the egg itself knows which is going to be anterior axis and which going to be posterior axis, because the head will develop towards the anterior side of the egg, and the abdomen will develop towards the posterior side of the egg. So those axes need to be determined already during the development, during the differentiation. So this becoid, it encodes a specific transcription factor, so becoid gene. Remember, gene we write in italics. This becoid mRNA is produced by the mother in the ovaries, but is not translated. So it is transported to the embryo as an mRNA, not as a protein. Okay, so this is transported to the embryo. Now the adapter proteins in the embryo recognize the 3' UTR of this mRNA, and they will transport this mRNA to one end of the egg. So here, if you look at this image, this is bicoid mRNA. So this is these proteins transport it towards the anterior pole. Now, there is another mRNA, nanos, another gene, and nanos. This nanos encode for an mRNA binding protein. The mRNA for this nanos from the mother is also transported to the future egg. It's transported to the posterior part of the egg. So here, where it shows Oscar mRNA, just think about it. Nanos will also be here. So becoid in the anterior, nanos in the posterior. So once the egg is laid, this becoid mRNA becomes activated. That means it becomes translated. It makes that specific transcription factor that it is destined to make. This becoid protein, this transcription factor, it diffuses through the cytoplasm. So remember, all the mRNA was in the anterior pole, at the anterior part of the mRNA. Now it starts diffusing through the cell, but this diffusion makes a gradient, because it's still, most of this will be concentrated towards the anterior pole, but then it starts diffusing, so it will make this gradient highest concentration here. As it moves towards posterior part, the concentration will be lower. So this makes this becoid gradient. These are the future. These are pole granules that will make that's where the pole cells will be formed on the posterior side. This becoid protein now binds with different affinity to the enhancer of two other genes, hunchback and orthodentical, in the local nuclei. So this is the hunchback expression. You can see the hunchback expression follows the pattern of bicoid expression as well. So more towards anterior pole and in the middle, and then it starts getting less concentrations towards the end. So its expression is mostly in the half of the embryo. Now, nanos protein gradient, remember nanos is from the mother, this goes on the posterior side. So here the nanos protein gradient is also higher concentration, is towards the posterior end, but then it diffuses, its concentration becomes lower, less and less as it moves towards the anterior side. So this hunchback mRNA is evenly distributed, though it's evenly distributed in the egg, but nanos protein, so this it looks like this because the nanos protein is in this posterior part, and the nanos protein is bind to hunchback and destabilizes it. So wherever nanos concentration is high, hunchback expression will be low. So that's why it's an opposite expression pattern appears. So this is the hunchback expression of mRNA before nanos protein gradient starts to destabilize this, and then once it starts to destabilize it, so it destabilizes where more of the nanos is present. So now the nuclei that express both orthodontical and hunchback, they will become the head structure. The one that only express hunchback, that will become thoracic structure. So this because this hunchback is more towards anterior, so this will make the head and thorax, and this portion will make more of the abdomen structure. Okay, so this is the head structure. That's where they both are present, or both orthodontical and hunchback, and then thorax will have less and less so this gradient will change as we move. Now, this is because Drosophila embryogenesis is very, very well studied. We know exactly all the stages of embryogenesis in Drosophila. So here, this is DAPI staining, pretty sure. So here it starts showing the first stage. There are just a few nuclei here. These nuclei start to divide more, and then these more nuclear division. As I mentioned, in Drosophila, as in most insects, the first many divisions are nuclear divisions only, and then once those nuclear divisions are there, they make this blastula. That's where when the cells will start moving towards the edges, towards the end here, and they will make their own cell membrane around those nucleus. So that's the blastula stage. So this is called synctium, and synctium is that's where all these nuclei are present without any cell membrane so far, and this keeps on dividing. So there are about 10 nuclear division post-fertilization happen first. So these makes about, if you go by every time, the cell, each cell will divide into 2. In 10 nuclear division, there will be about 1024 nuclei. This takes about 1 to 2 hours in Drosophila. Okay, so this is a fertilized egg. This is one nucleus. This is the diploid nucleus here. These are pole granules. These show that this is where the posterior end is. This nucleus then will start dividing, about 10 divisions, and this makes this many nuclei in a synctium, and that means there are no cell membranes yet. So the cell division hasn't happened yet. These nuclei start moving to cortex or periphery region. Here there'll be another 3 nuclear division that makes it about 6,000 nuclei within 2 to 3 hours of this embryonic development, and these nuclei migrate to periphery, or cell boundaries start to form. These nuclei are still totipotent. Here, this multinuclei cell before cell division is called the synctium. This is important because when we are making transgenic animals, when we are making transgenic Drosophila, we want to inject our materials during this when the synctium is when the egg is still in the syncytium formation, because the reason for that is there is no cell membrane around these nuclei. So if you inject something, there is potential of all of these nuclei to take up that material that you are adding in. Once the cell membranes are formed, then whatever you are injecting need to pass through the cell membrane, so it's harder. So that's why it says you have to determine for your organism when the cell is still without a cell membrane, when the embryo division is still without a cell membrane, if that happens. About 3 hours post-fertilization, the cellular division takes place. That makes the cellular blastoderm, the hollow ball of cells, or blastula. Now this is the cellular division is complete. They have these cell membranes. These cells will make somatic cells, and these cells around here with the pole granules will make pole cells. Pole cells are your germline cells. These are the cells that will give rise to the future ovaries or testes. So another thing, when you're making transgenic animals, so you want to know which end is the posterior end of your embryo. So you can inject close to the posterior end. That means you will be injecting close to these pole cells, close to these germ cells. So here, this is another a quick video showing different parts of the embryo development. So right here, this germ band formation starts. So this is the germ band. This germ band is now making the ventral furrow here. And here you can start seeing the segmentation. So the body segmentation has started. This right now is this is the ventral furrow is getting complete. So it's a germ band is complete at this point. All these different segmentation is complete, and now you will start seeing the limbs or appendages will start showing up soon. So here the cells are still moving. Germ band is now closed. The furrow that was made, the ventral furrow, that's now closed, and now it starts to look like more like a Drosophila larva. Here the little mouth part starts to form. And as I mentioned earlier, these very early embryonic development are quite similar in all organisms. Okay, so let's see this. Don't know what happened there, but that's okay. Let's move on. Okay. So there are some genes that are quite important developmental regulators. These are called homeotic genes. These homeotic genes, they bring this morphological diversity of different body segments. So these are very important for correct orientation, correct segmentation of the organism. So here in this example, we have, so this is a fly. So again, this is Drosophila, but think about all flies. And you will, when you think about insect, also think about some insects have 2 pairs of wings, so 4 wings like butterflies. Other insects have only 1 set of wings, 1 pair of wings, or 2 wings like flies. So the flies, Drosophila, mosquito, they all are flies, so they have only 1 pair of wings. So you will notice here that this pair of, this is a a wing pair here, where the other wing pair will be in the insects that have two sets of wings. Instead of that, flies have halters. These halters are the structure that are required for balancing when they're flying. These provide the balance to be in the air. These halters are part of the thorax. So all insects have three body parts: so head, thorax, and abdomen. This is also very important. So these homeotic genes, these regulators, they determine these different segments of the body. So during the segmentation, they would make sure which genes need to be expressed where and which need to be repressed in certain segments for the correct orientation. If this control, this regulation is not complete, is not controlled very well, it can affect the formation of these different body parts. For example, here in this fly, you'll see this is a head, thorax, and another thorax, and then abdomen. So this fly here has two sets of wing, because there's one set of wings is on this part of the thorax, and another set of wing is the second thorax part. This is the change in ultrabithorax expression. So there are several of these homeotic genes that are present in all organisms. So here, the best studied homeotic genes in Drosophila are Actinopedia and ultrabithorax. So this is an example of Actinopedia, and I'll show you in the next here this fly again. And this is an example of when the regulation is not controlled for ultrabithorax. So here, Actinopedia controls the development of middle segment or meso of thorax. So thorax has three segments: prothorax, mesothorax, and metathorax. Recall the flies, insects have three sets of legs. So here you can see they have three pairs of legs, total 6 legs. Each of these pair of legs will be on each of these segments: pro, meso, and metathorax. Metathorax also have this the halters. Actinopedia is not expressed in head, but if you forcibly express Actinopedia in the head, then there will be formation of these legs instead of antennae in the flies. So what that suggests is Actinopedia controls the leg formation on the thoracic segments in the flies. Ultrabithorax, this ultrabithorax gene encodes a homeodomain regulatory protein, and this homeodomain regulatory protein controls the development of metathorax. Metathorax is, see, that's where the wings are. So ultrabithorax expression, this represses the expression of genes required for development of mesothorax. So it will, it will repress the genes that require development of mesothorax in metathorax. Okay, so that means metathorax becomes metathorax. This ultrabithorax also represses Actinopedia expression, and it restricts this expression to mesothorax. So that way they can have the wings formation and legs formation there. If there is a misexpression of this Actinopedia genes and other gene regulated by this ultrabithorax, then the metathorax result in a duplicated mesothorax. So instead of a metathorax, there are two mesothorax. There are two mesothoraces here, and they both have their own set of wings there. So in flies, as I mentioned, mesothorax contains a pair of wings and legs, because each of those thoracic segments have one pair of legs, and metathorax contains the halters part of the fly. So here, this contains this halters. These halters are as I mentioned, the structure that help balance the insect during flight. This is important for flies because they don't have two pairs of wings for balance. So one of the pair of wings has become these halters and legs. So if there is a change in expression of ultrabiothorax, so that will change in multiple different genes, including antennapedia and other genes that are involved in formation of these different thoracic segments. And this will lead to, this can lead to a number of different changes in the segmentation pattern. And this is only one example of those different changes. So these mutant flies, these have they don't have their development very well. These flies don't survive either because they cannot fly very well, they cannot balance. The third way of differentiation is signal transduction, and as I mentioned, since we have already covered this, I'm not going to talk about this much, but think about it. Signal transduction is more of a distance regulation. So any molecule, a ligand, that ligand can be a small molecule, for example, an acetylcholine, dopamine, or it can be a peptide, for example, insulin, or it could be a hormone, again, a peptide hormone like insulin, or could be insulin growth factor, or it could be any other estrogen, for example, any hormone, a steroid hormone or a peptide hormone, a peptide or a small molecule. Any of these molecules, they can diffuse through the cytoplasm, that they can diffuse through the body, and they can find where their receptor is on the cell membrane, and here they bind to the cell membrane. Binding of this ligand to receptor then activates a number of different processes, including, if it's a tyrosine kinase, that will be phosphorylation. If it's a G-coupled protein receptor, then it will again lead to the activation of the G proteins. So either way, these binding of these ligand molecule to their receptor will activate the downstream processes, and these downstream processes, if it activates a transcription factor, then it will enter into the nucleus and bind to DNA, and it will activate transcription. If it's not a transcription factor, then it will lead to change in or activation of whatever gene that needs to be activated. So in the cytoplasm, it will go activate the transcription. And that's how the signal transduction works. So this is all we're going to cover. This was your last lecture, and again, this will be on the final. Final will be cumulative, so this will be throughout the semester, whatever we have talked about, including the SARS-CoV-2 lecture that we covered on Friday, and then this lecture today. I would like to meet with all of you face to face if possible on Thursday after the final. So I will be online if any of you, I will send you a link for a Zoom call. I will be online if any of you want to log in. I will be happy to hear from you. Okay, and with that, well, hope you have a good rest of your summer.